Hello, everyone. My name is Kevin. I am a member of the Evolution Seminar Series Committee. I want to welcome everyone here today to our Director's Lecture. We have a very special speaker today, but before we get started, does anyone have any announcements for the evolution community? Yes.
I was going to say that there's the Natural Selection Reading Group, which is going to be starting on Mondays at 11 next Monday. If you want to join that, contact Emma Howell or me, or if you're on the evolution news group list, you should be getting emails about that, so you can check your inbox. There's more information on the website, Evolution, Wisconsin.
Yeah, so feel free to check out the website if you're interested in our natural selections reading group. There's a fantastic paper being discussed on Monday, one that I have personally read and I think is really fantastic. But without further ado, I'd like to introduce our speaker coming all the way from Harvard University. We have Professor Robin Hopkins, who's going to tell us today about her work in plant speciation and adaptation. So welcome, Dr. Hopkins.
Thank you, guys. I am absolutely thrilled to be here. It is fun. This is my first seminar visit since COVID, and it's really special to be here with you guys out and about. And particular thanks to JoJo and Kevin for organizing this visit. Class I taught when I was at Harvard, and you all here and to be here. So thank you all. Yeah, so I'm surprisingly nervous. You don't do this for a few years. It's like, whoo, all right, we go. I'm really excited to share some of the work, a little bit of old stuff, some new stuff, some future stuff that we're working on, all thinking about the evolutionary forces driving speciation in Phlox, which is what my lab studies. So maybe we'll figure out how to use a computer. All right, so the lab studies speciation. And I'm just going to define speciation right now as the process where one interbreeding group of organisms splits into two distinct non-interbreeding groups of organisms, or less interbreeding groups of organisms, right? So this is an evolutionary process that takes place over time. And as someone who studies speciation, what I actually study is the evolution of reproductive isolation. So this is like a jargon term that refers to basically any trait or mechanism that stops or reduces the effective interbreeding between these populations. So it can be a trait like flower color, or it can be a trait like sperm inviability, but it's all of these traits that evolve within a population that stop them or reduce their ability to mate with other populations. Okay, so this is like the traits that we study to study speciation in that process. How did those traits evolve? And particularly, we study the fundamental evolutionary forces and how they're involved in the evolution of these reproductive isolating barriers. So we study selection, mutation, and migration, right? And what I hope I convince you of is that this lens of speciation, this lens of studying reproductive isolation, is actually a really cool lens to look at these pretty fundamental and universal processes of evolution in. So I'm going to walk through some vignettes about how we study selection, mutation, and migration in the context of this speciation. So let's start with selection. Let me introduce you to my little caricature flowers here. Most of the time when we think about selection and speciation, we think about ecological adaptation and sort of ecological speciation. So you might imagine there's a red species that is adapted to a red environment and a blue species adapted to a blue environment. And if they grow in the wrong environment, they don't do as well, they don't reproduce as well, and they die off. And if they do hybridize, that hybrid is sad and bad and does particularly horrible, and therefore is reproductively isolated. Those two species are reproductively isolated because of their differential adaptation. That's super cool. We do study that in the lab with these big reciprocal transplant experiments between closely related species to see how and why they're ecologically adapted. That's awesome. I'm not going to talk about it. Instead, I'm going to talk about this other process of selection that is really, I think, important for speciation, and that is reinforcement. Okay, so again, we're going to go back to characters to understand what this process looks like. Now we've got two blue species, big and little. If they share a pollinator, they may hybridize and produce sad hybrid, right? That sad hybrid is going to decrease the fitness of the parents and thus generate selection favoring any trait that reduces hybridization. So imagine now we evolve one of these, evolves a new trait that, say, attracts a different pollinator, that then these two species have different pollinators, they don't reproduce. That red flower in this case, that change in flower color, is under selection simply because it reduces hybridization. So in this way, selection favors the evolution of reproductive isolation because it actually causes reproductive isolation. So this is a distinct form of selection that can be really important for speciation. And it's what we spend a lot of time in our lab studying. So I'm going to dive into sort of how we study this process and the types of questions we ask and some of the cool data we've got. So what we're particularly interested in is understanding the cost of hybridization, right? So how bad is it to make sad face hybrid plant there? That is going to be really important for determining the strength of selection against hybridization, right? It's not so bad, who cares? The other important aspect that goes into determining the strength of selection is the amount of hybridization. If the species don't hybridize much, we don't need to stop it. If they hybridize a lot, that generates a lot of selection to reduce hybridization. And then the amount that any new trait, in this case, this change to a new pollinator, actually decreases hybridization is directly related to the strength of that selection, right? So these two aspects of understanding selection are really important for understanding how reinforcement works. All right, so we're going to dive into this looking at Phlox, which is, this is Phlox drummondii. It's an annual wildflower that grows through the roadsides and open fields in central and eastern Texas. Makes these huge, beautiful swaths of color. And it comes in these two flower colors. So one of them on the left over here, right, is sort of a purpley, light bluish color. It basically looks like every other phlox that is native to all around here. They all have this sort of bluey purple color. But this species also has evolved this deep dark red morphology. And this flower color transition was thought to have evolved because of reinforcement. So if we look, whoops, let's go the other way. Oh, somehow. There we go. All right, we're going that way. Okay, so this is a geographic distribution of the Phlox drummondii, where we see throughout most of its range in blue, we have this sort of light blue purple flower color. On the eastern edge of the range, all the plants have dark red flower color. And that area of dark red actually overlaps with this area of sympatry or geographic overlap with a closely related species, Cuspidata. So where you see red on this map, those two species co-occur. And this Cuspidata plant up there in the corner actually looks quite similar to the drummondii plant. And it was thought that maybe this evolution of flower color was driven by reinforcement to decrease hybridization in that field, in those sympatric populations. And so if we actually go to this area of the range, we can find that there are indeed populations that have drummondii up in the top corner, Cuspidata in the middle, and then hybrids stuck over there. And these hybrids are vigorous and awesome. So hybridization does happen. So in this system, what we want to ask is then, what is the cost of hybridization? Is there actually a force that would generate selection against hybridization? And then how much hybridization happens and how much does this shift in flower color change hybridization? And it's through this that we can understand the strength and dynamics of selection driving flower color evolution. So first, let's look at the cost of hybridization. So this was work done by a postdoc in my lab, Dr. Sevan Suni, a couple of years ago. And what she did is she took the two parent species, Cuspidata and drummondii, and crossed them together to generate hybrids. They actually cross pretty well. And those hybrids are big and vigorous. And actually, when we see them in the field, they look awesome. So we pretty quickly realized that this ecological selection against the hybrid was not going to be strong. And instead, we started to look at fertility of these hybrids. So what Sevon did at that point was then characterize the seed set. When you take the parents and you cross them to other of the parental species, these plants are a pain in the neck. They get about 1 to 2 seeds per cross. So you do a lot of crosses and you see that there's successful fertility and seed set. Now she can also then take the hybrids and back cross them to the parents and then cross them to each other to look at the fertility, both in pollen and ovule fertility of those hybrids. And it turns out it's really, really low. So depending on how you make the hybrid and who you back cross it to, that sterility or loss of fertility and incompatibility is incredibly high. From, you know, the best case scenario, about 50% seed set, and at the highest case, it's complete pollen sterility of an individual. And so what we see is that indeed that cost to hybridization in this case is really big. And it's not that those hybrids do poorly ecologically, it's that they don't make babies, right? So that's a pretty big evolutionary dead end. So we have a potential for really strong selection to stop this cost. But then we might want to know, of course, how much hybridization actually happens. And for this is some cool old work that we did, actually measuring hybridization in the field. So this is Drummondii and Cuspidata, which grew out in the field together. And the Drummondii, we took the allopatric ancestral color, right? So before it evolved dark red flower color, how much hybridization would have happened if the flowers are actually the same color put together with the pollinators? So we grow out these huge patches of these plants, we let the pollinators do their business, and then we come back, collect their seeds and see who the dads are. So from these Drummondii individuals, we did paternity testing to see if they were hybrids with Cuspidata or they were purebreds with other Drummondii dads. And what we found was nearly 40% of these offspring were hybrids. That's a bummer, right? Like half their offspring are sterile at an evolutionary dead end. That could be really, really, really strong selection. So now we've set ourselves up for the potential for super strong selection favoring a reduction in hybridization. But did it? Right? Did this transition from blue to red, this like purpley color to this dark red color actually decrease hybridization and therefore be driven by reinforcing selection? So the amount that we decrease hybridization is going to be super important for understanding the dynamics of selection there. So before I tell you the answer, we're going to dive into a little bit to what this flower color change is. So these two flower colors are made from producing anthocyanin pigments. These are floral pigments made in many, many different kinds of plants and flowers. And what we notice is that the difference between these two are both sort of the amount of pigment made and the types of pigment made. So if we cross the two within species differences, we can make a hybrid F1 that produces a lot of pigment and is kind of this purpley dark blue color. We then self that and we segregate 4 distinct flower colors. We get the parents, light blue and dark red. We get the F1 color, dark blue, and we get this new sort of light red, pinky color that's quite pretty. And we can quantify this flower color on a 3-dimensional color space using a spectrophotometer to get really good quantification of spectral reflectance. And when we group these colors, we see that there is really distinct grouping. And across different F2 populations, we can explain, 75 to 85% of the variance with these categories, which is pretty cool. What's also cool is that we get these 4 distinct colors in a very pretty 9:3 to 3:1 ratio. Thank you, Mendel. So this was a glorious point in my PhD career where I was like, I know what that means. That means there are 2 loci with 2 alleles and complete dominance. It turns out one of those loci controls the amount of pigment made. We call this the intensity locus. So you start with low intensity, little I, and they evolved high intensity, high pigment production, big I allele. The second locus controls the type of pigments made. So the ancestral state makes both blue and red pigment. You get this bluey color. So this is the hue locus, You start with little h. You then evolve into this derived allele, which only makes, or phenotype, which only makes red pigment. So then you go from big H to little h, from making blue and red pigment to no longer making blue and only making red pigment. So we've got the hue locus and the intensity locus. This color can be decomposed into two aspects, intensity and hue change. Great. But what we really want to know is, does that change in intensity or hue actually change hybridization? So we throw them all back out in the field, right? So I didn't tell you, we did it all at the same time. We threw out the light reds and the dark reds and the dark blues and the light blues in the field with a bunch of cuspidata, that's that other species. We let the pollinators do their song and dance. We go back, we collect the seeds, and we ask who the dads are. And in this way, we can estimate the hybridization rate of each of these flower color morphs and see if changing the intensity or changing the hue actually changes hybridization, and therefore, demonstrates how that reinforcing selection actually works. Well, I wouldn't be up here if it didn't work. So what we found was that the change in intensity, this shift from light to dark, from making a whole bunch of a little pigment to a whole bunch of pigment, actually decreases hybridization by 50%. So you went from 40% of the offspring being hybrids to 15 to 20% of the offspring being hybrids. That's huge, right? And if we think about then that strength of selection to decrease hybridization by 50% when hybrids are sterile, that gives us a pretty good idea that selection was probably pretty strong. So cost of hybridization was big, the amount of hybridization was a lot, and the decrease in hybridization was a lot. We have super strong selection for speciation. How? Right? I don't know about you, but I'm like, okay, that's magic. That's crazy, right? You change intensity of a flower pigment. How do you actually decrease hybridization? So of course we had to start looking at the butterflies. So this Phlox in general has a very characteristic lepidopteran pollination system. They've got these long corolla tubes that perfectly fit the nice skinny proboscis of a butterfly or moth. Very end is a little drop of nectar that they love, right? So we sort of went in knowing that there was going to be butterflies that are pollinating it, but which butterflies and how many? So this is some recent work done by an awesome graduate student in my lab, Grace Bergen, who went out and spent 55 hours watching. She went across a bunch of sites in Texas throughout the course of the day, watched over 3,000 pollinator visits or flower visits to different plants, and well, pretty quickly realized that it's this one pollinator. This pipevine swallowtail, Batis philonor, is an incredibly common pollinator as butterfly out there. And it's really, really good at pollinating phlox. It visits, it collects a ton of pollen, it deposits a ton of pollen, and really good at allowing seed set when depositing pollen on these phlox. So this guy is our major pollinator. It's actually the female butterflies. This lady is our very good pollinator with over 80% of the visits and incredibly effective pollination. All right, that's great, but how does it differentiate or change with these light blue or light flowers versus dark flowers? They go to everything, right? They're the major pollinator in the light blue region. They're the major pollinator in the dark red region. They pollinate cuspidata like it's all Batis all the time. So what we did then was put out little potted plant gardens of the different colors and the different species and sat and watched how these Batis moved between them, right? How are they actually moving pollen between species and between colors, and does color affect the way they move between species? So we looked at the lights versus the darks and the reds versus the blues. All right, so I'm going to show you the data like this. Basically, if a butterfly is on a light blue Drummondii, 28% of the time, its next plant that it went to was a cuspidata. And if we observed a pollinator on the light red plant, 24% of the time, the next plant it went to was a cuspidata, right? So this is the proportion of transitions between species depending on the color. And they're the same, whether they're light red or light blue. And if the butterfly is on a cuspidata, again, the same percent transitions over to Drummondii regardless of that color change. But when the Drummondii are dark, the pollinators differentiate. They still go to the darks and they still go to the lights, but they don't go between the dark Drummondii and Cuspidata. They go between the darks and then they go between the lights and not between the two. And the same is true for the dark red. In these gardens, we found that they went from a dark red to a dark red, or from a light blue to a light blue and a Cuspidata. So this is a pattern of what we call constancy, where butterflies basically get a search image of what they're going to go to. And some individuals are going dark to dark to dark to dark, here red, and other individuals are going light to light to light to light. And in doing so, they move pollen between species that both have light flowers, right? And so this pattern of pollinator movement actually matches really nicely with this pattern of hybrid seed set that we saw in the field. So if we go back to thinking about selection driving reproductive isolation, through this process of reinforcement, we can see there's a really big cost to hybridization that has the potential to generate selection, and that hybridization is high with a huge decrease in hybridization caused by this simple trait of flower color change due to pollinator movement or manipulating these Batis butterflies, which is pretty awesome. All right, so that's sort of one way we can start thinking about the evolutionary process of speciation. The other way, the next way that our lab focuses on studying speciation is thinking about the mutational basis of speciation, right? What is the raw material that evolution uses to actually get the evolution of reproductive isolation? What are the mutations? Maybe just genes. All right, we talked a little bit about this, right? Some classic genetic crosses allowed us to say that there are two loci, each with two alleles and complete dominance, right? So we want to know what's the hue locus. It changes from this blue pigment to making only red pigment. And what's the intensity locus from making a little pigment to a lot of pigment? What is that? So we got excited about diving into the intensity locus because that's the one that we could really see an effect on behavior in, right? So we'll go there, and plus it's easier to look at. That's the deep dark secret I'll talk about later. All right, so as I said, this intensity locus, well, all of these color changes are due to changes in amount and type of anthocyanin pigments. Anthocyanins are the biosynthetic pathway to generate anthocyanin pigments are conserved across plants. And this is it in basic schematic, where these bold enzymes next to the arrows are basically genes that you can find in any plant, right? The primers work for any of them. It's beautiful. And then in the box, there's transcription factors that have been shown in a wide variety of plants to regulate these core enzymes in this pathway or the expression of these genes. All right, so what can we do? We have these F2 populations that segregate flower color. We can then go into our plant genome and actually sequence these candidate genes, right? Chances are if you're affecting pigment, it's gonna be one of these genes. We've sequenced these genes, looked for different SNP variants in the different parents, and then looked for a genetic association between any of these core enzymes or these transcription factors that are associated with these changes in flower color, right? That are different between light and dark or different between reds and blues. So for light and dark, we found a perfect association with this transcription factor. This is an R2-R3 MIB transcription factor that, you know, is one allele in all the lights and one allele in all the darks in our F2 population. And what's cool, I mean, it makes perfect sense. In other plants, this type of transcription factor has been shown to regulate all the expression of the genes in those anthocyanin pathways. So sure, that could very well influence the amount of pigment that's being made. But let's dive into the function of this gene, right? Can we actually validate that this MIB is doing what we think it's doing, that is regulating the amount of pigment in a flower in our phlox? Because our phlox are kind of funny. All right, so the first thing we did was actually just a classic gene expression assay where we look at the transcript levels in the dark individuals and the light individuals. And we can do this in F2s and we can do this in natural populations. And in every case, we find that dark individuals make a ton more transcripts than light individuals of this R2, R3 mid. So we have higher expression of dark than light in the flower buds that are making the pigments of this gene. We can also look in the F1s, right? So this is an individual that we know has one light allele and one dark allele. This is genetic DNA composition of 50-50, right? A light and a dark. We can look at the transcript abundance of the light and the dark allele in those F1s. Yeah, and we actually find that in those F1s, the dark allele is overabundant. So the allele-specific expression also shows that that dark allele is pumping out transcripts way more than the light allele, consistent with an upregulation of gene expression of that particular dark allele. All right, so we go back to our color space and we have these 4 flower colors, and our hypothesis is that we're going from dark to light by changing expression of this R2, R3 mid transcription factor, okay? How does this gene, could this gene actually do that, right? How can we actually functionally validate that this gene is capable of making this shift? So to do this, we use a fun little trick in plants. This is work done by fabulous graduate student Austin Garner in the lab. And he does this transient gene silencing technique called viral induced gene silencing, or VIGS. It's a very cool plant trick. You think about within a cell, this gene, this R2, R3 mid gene, is being transcribed and then translated, made into this transcription factor that upregulates all the genes. This virus-induced gene silencing, VIGS, actually infects a plant with a virus that expresses a little portion of the gene you're interested in silencing. And it makes a sense and an antisense copy that then form a double-stranded RNA molecule. Anyone who knows RNAi knows that that double-stranded RNA molecule will then trigger a degradation RNAi endogenous mechanism to chop up those transcripts. This targets both the viral-induced RNA expression, but also the endogenous copy. So in this way, you can get the plant itself to silence a gene that you're interested in. So we literally infect a plant with the virus, trigger its immune system to chop up the endogenous copy of a gene that you're interested in. And in that way, we can actually reduce the expression of this R2, R3 mid transcription factor and see if we can recreate this light flower color. We can, it's so cool. So the really cool thing about these VIGS experiments is that they're mosaic. So you get some branches that are uninfected and don't have silencing and remain super, super dark. And some branches that you really effectively infect and silence or decrease the expression of that mid. And sure enough, you get these super light flowers that pretty clearly match what we see in the field as the sort of natural color of the light plants. So we can look more carefully at this in our color space. We actually can show that on the uninfected, unsilenced side of a plant, we're dark, and the light infected side flowers, we actually get a shift in the axes that we would predict. If we lower expression, then we lower this color. And so sure enough, what we can show using qPCR is that we have effectively silenced this gene in these flower buds, and that pretty much recreates that silencing that we see in nature, the difference in expression that we see in nature. All right, this is pretty cool. Like, we're super excited that we can demonstrate that this is the gene that could cause shifts in flower color intensity. That doesn't tell us that it did, right? So what we want to know now is, did it? Are there actually mutations that control the expression of this gene that actually increase expression of the R2, R3 mid transcription factor? All right. Here's the little dirty secret about Phlox. It is a giant genome. So we've got 6 gigabases of a genome spread over 7 enormous chromosomes. And somewhere hiding in there is our R2, R3 mid transcription factor. And we think, based on other studies of flower color, that if it is a cis-regulatory region, right, something that's close to that particular gene or allele that's controlling expression, it's going to be within about 5,000 base pairs. So what we want is like a little, snippet of the genome. Like, I don't care about the rest of the 6 gigabases. That's really, really, really hard to look at in an F2 population, right? A 6 gigabase genome or 7 chromosomes, how many F2s would you need to find map that? Like, I don't even want to think about the greenhouse space. We'd need like greenhouses all over the world to get enough plants to find map that. So instead, what we were thinking about is actually going to these regions of natural admixture. So in nature, right on this edge of where the light blue and the dark red are, there's plenty of populations with all the colors. And they segregate, and they have been segregating for many, many, many years, and there's many, many, many individuals. And so we can get genomes that are very mixed up, and then look in them and compare lights to darks and see if we can narrow in on an association between particular variants and the intensity locus. So use natural admixture to do association mapping. Okay, so now all we need to do is zoom into this teeny region of a 6 gigabase genome, sequence it out of tons of individuals, and try to find a variant. Oh, good Lord, this is not a model system at all. So how do we do that? This was some awesome sleuthing that this graduate student, Austin Garner, found. So this company called Samplex uses this pretty cool extra nanopore sequencing technology where you can take a single probe in a gene. So for us, we take this R2R3 mib gene, coding region of the gene, we put a single probe in. We use long, high molecular weight DNA. We pull out or we isolate any long strands of DNA that contain that probe, isolate them into little droplets, and then use long read sequencing to sequence those long strands of DNA. And in this, using this method, we actually get about 80,000 base pairs of sequencing right around the coding region of our gene. So what we can do is now exactly what we dreamed of, zoom in and target this gene, but sequence around it far enough out that we're confident we would most likely get the signal of association with a causal mutation. And the great thing about this technique is that we have high coverage long read data. So we can resolve point mutations because we have such high coverage, insertions, deletions, duplications, and rearrangements because we have this long read data that give us really nice resolution of structural variation. All right, that seems like magic, but we give it a try. We started with just 81. Now we're up to, I think, 120 individuals. Those data are being analyzed of all different colors. So we sort them by light and dark. We sequence them. We use a simple GWAS analyses. Boom. And we find an association with 2 SNPs. That's it. Thousands of variants across 80,000 base pairs and 2 SNPs. All right. Both of them lie right upstream of the first exon of this gene. So shown here are the 3 exons of the R2R3 mib transcription factor. And we see that these 2 associated SNPs are right next door. And they are this top one, G to T, is perfectly associated with light to dark. And when we quantify brightness using brightness and chroma, we see this really strong association between genotype at that SNP and the color of the flower. And importantly, it's not associated with anything else around it. So what are these SNPs? Yeah, good question. So we dive in a little bit closer and we can characterize we can infer some of the function of the promoter region around here. And we see indeed they're within 200 base pairs of the start codon, and what they are is right in between the tata box and the cat box. So what this makes us believe is that this is in the core regulatory promoter region, right? So it is involved somehow in attracting and function of some of those core regulatory proteins. Exactly how, we don't know yet. So our future work is to actually characterize the effect of these mutations on promoter function, but we'll get there. Either way, I think it's pretty clear that we are able in this totally non-model organism to start diving in and learning something about the mutations underlying reproductive isolation and speciation. So we can combine classical genetics to get our gene candidate genes and sort of a general idea of what's underlying them, then use some functional validation to start untangling sort of how these genetic mutations might work. And then this targeted deep dive association mapping to actually narrow it down now to what looks like 2 SNPs that could possibly be underlying this change in flower color intensity. Pretty fun. All right, we got one more evolutionary force to go look at. Here we go. Migration. Why do we care about migration or gene flow in speciation? I don't know about you guys, but do you ever look through like table of contents or pops up on your Google Scholar, whatever? Like every other paper, maybe this is just me, every other paper is like history of gene flow, history of admixture, hybridization is everywhere. Like in the speciation world, this is taking over. Everybody is like crazy excited about hybridization in history. I mean, so am I. But I wanted to step back and ask, like, why do we care, right? Why does it matter if there has been hybridization or gene flow in the past? So let's think about this. We're back to some flowers now, they're yellow, right? And there's the big and the little species, and they're growing together and they're making sad hybrids. That's what you're looking at here, right? Now let's imagine that you evolve this new fabulous trait and it stops hybridizing. This trait spreads. Now you have 2 coexisting species that aren't hybridizing. That's better, right? Speciation. What if there's gene flow? What if there is migration now during the evolution of this trait, red flower color? Now we have a hybrid that's carrying that red allele, right? And it can act as a conduit to move it into the other species. Well, now the other species is red too. And guess what? It hybridizes like crazy. And all of a sudden you're back to losing this distinction between these species. And again, you have a hybrid swarm of a big and a little species. Now they're red and they've lost. It is more difficult to maintain their co-adapted gene complexes and genetic identities, right? So that's it. That's the reason why people are so excited about studying a history of gene flow. If you see a history of gene flow between 2 what are now good species, you know that somehow selection or whatever was able to drive the species apart despite this force of homogenization that happened. All right, hopefully that wasn't too obvious for you all, but I think it's important to kind of go back and think like, why do I care? It's because of that. All right, but why do we care? We want to know if there's evidence that this dark red flower color in Sympatry evolved despite gene flow between them. Was that homogenizing force in evolution countering the divergence that was happening between these 2 species? So we're going to take a sort of phylogenomic perspective. Here is a snippet of a phylogeny, schematic of a phylogeny. No details of this phylogeny. Besides, in the top, you see Drummondii, light in blue. Sister to it is actually another species that grows in Texas, Phlox mariana. And then out to that is this cuspidata species that we've been interested in. Out to that is another species that doesn't grow in Texas, Phlox pilosa. Okay, so but what we're interested in is gene flow between this red Drummondii and cuspidata. Cool, not its sister species, but still pretty close. They're only all like those 3 are, 2 million years old. They're pretty young. So when we sequence transcriptomes and we do whole genome dd-rad, reduced representation genotyping, all of our estimates of species trees give us this topology, right? So there's a lot of gene trees and a lot of SNP topologies that are concordant with this. Drummondii, sister to Ramariana, out is cuspidata, but not all of them, right? And we can use these patterns of discordance, or I don't show this topology, to infer if there has been gene flow between cuspidata and Drummondii, particularly these regions in Sympatry, which I'm denoting as the dark red ones, right? So let's break this down just a little bit. Here's what a gene tree or a SNP topology might look like that is concordant with the species tree. As we move forward in evolutionary time from an ancestor up, we might see there's ancestral alleles, and then we evolve some new allele B, and everything after that that is evolves has this B allele, this derived allele, right? So those 2 sister species both contain the derived allele. But you can get discordant patterns of SNP topology if you have incomplete lineage sorting. So what does that look like? That just looks like a new allele arises sometime in evolutionary history. And guess what? It doesn't like that, right? It segregates in a population. These populations can speciate, bring those segregating alleles with them. And by chance, one allele fixes or the other allele fixes, and you get these discordant patterns where the Bs are in non-sister species and the As are not all in the ancestral, the more anciently diverged lineages. Okay. The important thing about this is that it is kind of random. So you expect these patterns of ABBA and BABA, the ABBA-BABA test, to happen at equal frequency, right? About. There's nothing that says that the ABBA should go one way and the BABA should go the other way. It just sort of happens. So we see a ton of this in these Phlox genomes. And most of it is probably because these are young species and they speciated fast. So there was a ton of variation that all kind of got split and then sorted itself later on. Gene flow is subtly distinct from that. In this case, we would imagine a scenario where a B allele evolves on one lineage, and then by gene flow, it moves into the non-sister species. Now, what's important about this is that it will only show one of the two patterns, right? So in this case, we get only ABBAs from gene flow between these two lineages. So if we see a lot of ABBAs and less BABAs, we can infer that this is consistent with gene flow happening between those two lineages. ABBA, BABA, now we're the 70s dance party, but let's go back to Texas Phlox. Okay, so here we're just going to simplify things and look at those sympatric samples. This was some recent work done by Austin Garner again. He sampled 6 red individuals from sympatry, 3 cuspidata individuals, some outgroups, Romariana and Pelosa as well. All right, and he's looking for, most of them are showing this BBAA, SNP topology, and he's looking at ABBAs and BABAs and trying to see, all right, do we see more, in this case, ABBAs and BABAs consistent with gene flow between Drummondii red and cuspidata? And sure enough, he did. So because of all the samples he uses, he did about 48 tests. All of them had negative D statistics indicating in all of them, the ABBAs were more than the BABAs, and half of them were highly significant. So this gives us pretty strong evidence that there was some gene flow between cuspidata and Drummondii in this region of sympatry. What's kind of cool is that he also did it with individuals from allopatry that are these light blue individuals way over there, geographically distant from cuspidata. They show no signal of gene flow. So we see mostly negative, but no significant enrichment of the ABBAs compared to the BABAs. So this gives us pretty strong evidence that, well, maybe logically, the gene flow is happening in sympatry. It doesn't happen so well when you're 100 miles apart. Reproduction is hard then. All right, so he also used this model-based program called TreeMix, which infers the splits between lineages. So here's sort of its topology of splits, which perfectly matches all the other topologies that we ever get, which is Drummondii, sister to Romariana, cuspidata out. And then it infers migration branches between these lineages. And sure enough, we get a pretty strong migration branch coming from cuspidata into Drummondii and specifically into those dark red lineages, which is super cool. All right, so in the end, this just tells us there's gene flow. There was the potential, and maybe still is the potential, for these dark alleles to be moving into the cuspidata species. Interesting though, gene flow happened in the other direction. What we actually see is that gene flow is happening from the cuspidata alleles into the Drummondii, much more than the other way around. And maybe that's why we haven't actually seen the sharing of this dark plant, this dark allele into the other one. Who knows? It's interesting, more to do. But what Austin decided to do with this information is to step back and think about this analysis in the context of the Phlox phylogeny. Don't read this. This is a rainbow, okay? That's all you need to know. These are all the different colors or different Phlox species that are in this sort of eastern clade of all tall Phlox. So he resolved the phylogeny of this Phlox clade. And what's really cool about this group of Phlox, this is where our Drummondii is. And this is where this example of reinforcement is in the phylogeny. There's another example of reinforcement up there in the phylogeny, another color shift. Maybe there was hybridization that drove it, and maybe there was gene flow that's involved in it. There's 4 examples of hybrid polyploid speciation in this little group of like 20 species. And there are 5 hypothesized examples of homoploid hybrid speciation. So this is diploids that hybridize and form a 3rd diploid lineage. That's a mess, right? So this is like tons of hypothesized hybridization that was incredibly important for the evolution of this clade of plants. Well, we can use, Austin used, not we, Austin, use these techniques of looking at discordance patterns of gene trees and SNP topology to actually test if gene flow was involved in all of these. Like, is that right? That this is all due to hybridization and a chance for gene flow here. And so, as I said, he did find strong evidence of gene flow in this one case of reinforcement, but not the other, which is cool. So one of the things we want to study in the lab is comparing these 2 cases, right? One of them happened with evidence of gene flow. One of them, it looks like gene flow had stopped before we see the evolution of this flower color chain. It's a pretty neat scenario to start studying. We know that these are hybrid polyploids, and so indeed he could confirm some that they were hybrid polyploids and then infer some of the ancestry. And actually, in some of these analyses, the parents are not what we thought they were based on morphology and where they grow. They actually have a different genetic ancestry than we thought before, which is also really cool to think about, who are the parents of these polyploids. And of the 5 hypothesized cases of hybrid speciation that are homoploid hybrid speciation, we only actually found genetic evidence for one of them, right? And so it looks like this one lineage actually evolved with some gene flow between 2 hypothesized parents. The extent of that we still need to study and sort of what the context is for speciation. But this is really exciting to us because it does mean that this clade is chock full of cool examples of hybridization and gene flow involved in speciation, but maybe not quite as full as we originally thought based on some sort of historic morphological analyses in this group. All right, so here we are. It's almost 1:30. You done with me yet? I hope today I've shown you that we can actually do a lot to understand selection and its role in speciation with combining these greenhouse experiments with some in-depth field experiments to understand the mechanisms of selection and the strength of selection that's driving this process. Our future work is to incorporate some of our genetic investigations to actually look at the genome for signatures of selection and move this work forward to say, okay, either ecological selection or some of these reinforcing selections, do we actually see signatures, these classic signatures of sweeps that you might predict in loci that are under strong reinforcing selection. I've also shown you that we can combine some really cool classic genetics like Mendel-style crosses, so fun. Everyone in the lab hates them. Crossing them is a pain in the neck. With some really awesome sort of advanced molecular evolution, molecular function studies to really dig down into what is the molecular basis of these traits that we're interested in. And our future work right now that we're developing are these promoter functional assays. So we're using a dual luciferase promoter function assay to actually test how much does the light promoter drive expression and how much does a dark promoter drive expression of a reporter gene like luciferase in the flower buds. So that's to come soon, hopefully. Got the protocol working. Now we got to actually figure out the results. All right. And then, of course, understanding migration. And for this, we're doing a population genetic approach. And the exciting new advancement in this is that we have just sequenced and assembled the genome of Phlox. And so version 1.0 now has chromosome level assembly of the Phlox genome, which will allow us then to dive into thinking about gene flow across the chromosome and not just across geographic space. So that's some future work that the lab is really excited to dive into. Are there regions where we see lots of integration across lots of individuals and regions that we see never signs of integration from cuspidata into Drummondii? So with that, a huge thanks to my awesome lab, tons of people to thank, but mostly Sevan, Suni, Graceberg, and Austin Garner, Andrea Berardi, who's been helping a ton with this transgenic work that we're starting to work on, and a bunch of the technicians that have worked with me over the years. All right, we got lots of time for questions, so thank you, thank you.
We have a lot of people in the audience. There's the from class, so I'm going to ask my class a whole question and go into the other room so that people can have an opportunity.
Awesome. Yeah.
Thanks for the amazing talk.
Thanks for having me.
Very fun. I wanted to ask in Phlox, why out of that comparison? You're talking about Homoplied hybrids.
Yeah.
You're saying one of them had evidence of that, the other did not.
Yeah.
Do they seem to be just lineages that were otherwise doing faster evolution, or was there reinforcement there, or does anything know yet?
This is so cool. So this paper is hopefully coming out soon, submitted. So it depends on the lineage. So yeah, so there's these 5 examples of Homoplied hybrid speciation. One of them we show evidence is there's at least some genetic makeup from both of the hypothesized parents. That's one. Then 3 of them look like they're just, yeah, like some populations of another species, one of the parents, hypothesized parents. And what's interesting is now what we're starting to look at and think about is this morphological evolution, right? They share traits which seem to be from another species that's closely related, but there's no evidence that they got those traits from that species. So what is it about that morphological evolution that's different than the genetic evolution that we're seeing? And so we don't know, but for 3 of those cases, that's exactly what we see, where it's like particularly strange diverged populations. But when we look at like FST and whatever, it all just looks like within population variation genetically, but morphologically, they're really distinct. The last one is totally crazy. It's not closely related to either of the parents. It's like way out there and something else completely. And who knows why it was thought to be a hybrid. I mean, yeah, I mean, I do know why, because there was lots of morphological data that said it should be a hybrid, traits from both of these other lineages. There was some protein assays. So this was like studied in the 60s and 70s, right? So they did some of these really early protein assays to look, and they show protein compositions that were sort of heterozygous, or whatever that is, compounds from both. And microsatellite data said that they could be hybrids. But genomic data says that they're like actually distantly related to all of them. So it really depends. And I think it's really interesting to think about our assumptions of morphological evidence versus the rules we know genetics evolves, right? Like we know how genetic variation evolves. We don't know how morphological variation evolves. Yeah. I don't know if that answered your question, but it's totally fascinating for me.
Yeah, super cool. I was wondering if you know anything about, so you said the species were like 2 million years or around there. Do you know anything about the time scales of one, the new and the darker color, how long it's been around, and also anything about how long have they always been, has there always been this hybrid zone that they've been in contact with? I'm wondering if, if you have hybrids from where they're allopatric forming or having different levels of, sterility or offspring, but whatever, than the ones that are in the hybrid zone.
Oh my God, I want to know that. I'm going to do another PhD in my lab, postdoc, come figure it out. Okay, great. We don't know, but we're really excited. I feel like the genomic resources we have now can actually help us answer some of those questions. So I think you bring up a really interesting point that these patterns of gene flow are overlapping with geography and then demography of each of the parent species, right? So it's like a little bit of a race. Is it that hybridization is rare and localized, or is it that migration into that region was recent and everything is just new? And we don't know yet. And so, yes, there's actually a postdoc who's working on the genome is that's what he wants to do. He's like, I'm really excited to look into sort of the demography of these two species and compare that to the I don't know what you want to call it, like selection and hybridization demography overlaid on top of it, right? So we don't know at all. I could hand wave some more, but yeah, it's a really great question. But repeatedly we find, like when we did some transcriptome sequencing, again, we find no evidence of gene flow from individuals that an individual we sequenced from allopatry, but pretty clear evidence in sympatry. The weird thing is that there's really, and we're getting more resolution on this now, but there seems to be pretty weak, significant, but pretty weak isolation by distance. So that implies that you have really big effective population size and probably a lot of gene flow across space. So why then that signature of hybridization would stay localized? Maybe it's because it's recent, I don't know. Yeah.
Super exciting thought. Thank
Thank
you.
you.
I have this technical question about the trimmings. So I wonder how is the number of migration events determined? I think that the trimmings that we would eventually affect the migration events that you got.
Yeah, so when we ran tree mix, we run it with none, 1, 2, 3, 4, 5, 6, whatever. Well, I don't think there are even 6 possible ways to do that. So you run it with a bunch, and then you just ask sort of which one optimizes the, which one is the, yeah, exactly. And so what we find is that no matter how many edges you put on, that edge is always there. And this other edge that was, I think it's between Drummondii and Pilosa, that's sometimes there. And if you add more edges, it basically disintegrates. Like it can't figure out what to do. It just sort of starts sticking them on randomly at really low edges. So there's, yes, some variation in what you might get depending on what the edges are, but it was pretty clear in this system that that's sort of.
The issue is that whenever you increase the number of migration events, you always decrease the residuals.
Right. Right.
How do you know where?
Well, I mean, and the thing with what we were doing is a specific hypothesis test, right? So what we really wanted to know is, do we ever see one between red and cuspidata? And do we ever see one between blue and cuspidata? And the truth was we always found one between red and cuspidata, and we never found one between blue and cuspidata. So in that case, it didn't matter. Like maybe there are other edges that would come and go or whatever, but yeah, I mean, yeah. You're always trying to infer reality from a couple snips, right? So who knows what the reality is? I'd love to just go back in time. We're pretty confident in this, combined with some of these other tests. It feels good, but you're absolutely right. I mean, we're constrained by the statistics that we can do. Yeah, no, you're true. So true.
I'm curious if you try to combine some of the information you have about the obvious Mendelian inheritance of the colors, presumably you can score the frequency of each color in the hybrid population, and then you also know about some assorted mating kind of patterns between the colors. Does that, is there predictive value there? Can you say why the frequencies are what they are?
Yeah, that's actually a really interesting question. And what becomes tricky is the biology. That's not always the case, right? So here's the deal. In the pot, so anywhere that cuspidata is, Drummondii is dark red, always. There's like no variation in flower color. That change from dark red to light blue happens over about 7 kilometers. I used to joke, I was like, you could walk that, no problem. I never have. Austin, this graduate student, actually went out in the field last year and he walked it. And I get a call from the end of the transaction. He's like, this was a long walk when it's 100 degrees in Austin. I was like, yeah, but you did it. So I can still say it. I mean, it's like a super short, narrow zone, right? There's no cuspidata around. So the hybridization we're seeing, the mixing of alleles is all within species. What's tricky is that we actually don't see assortative mating within species by color. We only see it between species. So what this means is that the pollinators differentiate between species when they're different colors, but that color is not enough to cause this assortative mating. So the other species differ in other things too. They're slightly smaller flowers, they smell different, they sort of have different heights. And so it seems like it requires this combination of events that color is necessary, but without those other differences, they just move between them. So we actually see pretty random mating, what looks random mating in these mixed populations of Drummondii. And that seems, our hypothesis is that it's more influenced by like how much is coming from a light blue and how much is coming from a dark red just by movement. But we now have the sampling and the genetic data like off the sequencer to look more carefully at patterns of gene flow and see if that can help us predict sort of the steepness of the climb and kind of stuff.
Well, I was sort of almost wondering if the frequencies of color would like infer the gene flow. There's only certain rates of gene flow.
Yeah, but the decline of color is really removed from the decline of hybridization. Like there's a pretty big buffer zone there, we think, which is unfortunate because that would have been a lot easier to do. This color decline is bizarrely narrow. Yeah, it seems like there's no way selection could be that strong. But well, who knows? Good question though. I wish they would line up and we could actually use it. Yeah. Cool. Awesome. Yeah, more. I mean, I'll stay here forever. I don't know if the class needs to leave, but well, I'm not happy. It's so much fun. It's been like 3 years. I'm like, oh my God, let's keep talking.
Kind of piggybacking off of that, talking about the scale of that, of where that zone ends, do you know anything about the sort of movement range of the pollinators? Like, are they can find themselves, like a very small, only pollinating bottom of a certain region? Are they moving all the way across?
I love you. That's a great question. Oh, so no, we don't. These are big butterflies, and all we know is that you can't follow them. Like they fly up and they're gone. So this graduate student who did all this pollination work, figuring out that Batis is the main pollinator and that it's super effective, has this amazing project that's mid-go now, where she has population genetic sampling from across the range of Drummondii, so like 300 individuals. And then at those same populations, she collected the Batis, so she has like 250 Batis individuals. And so she's comparing the population genetics of the pollinator and the plant to try to get at some of these questions, right? Like that's not a direct measure of migration, but you can't run after these guys. So it's sort of as close as we get to like how much could they be moving? And does it line up with sort of their patterns of structure and the plant pattern of structure? So I don't know, what should we give her, like 6 months? And then she just got the data, so we're starting to dive in. But that's a great question. We really want to know.
GPS tagging butterflies.
We are not GPS tagging butterflies. I think all that's saved for the monarchs. No, these guys are pretty dangerous.
Now you want to see. Hey, let's think around.